Good morning, class, and welcome to today's activity where I'm going to go over the second part of our gel electrophoresis virtual lab. This one is more of a crime scene investigation, kind of a DNA fingerprinting lab. So guys, I'm going to run through this tutorial with you. Once again, for those of you who have issues with Flash, not being able to be run through your computer, guys, follow along this video. Pause the video when you have to write some answers. We're going to be filling in a virtual notebook, and you could record your answers on a separate sheet of paper since you won't be able to have this access to your computer. So guys, let's go ahead and begin. As soon as I click this, you're going to start to hear some audio. So we're going to do gel electrophoresis.
A local neighborhood has been the scene of several recent bicycle thefts. A mountain bike has just been stolen, and two suspects have been identified. Fortunately, a hair was found on the broken bicycle chain at the crime scene. In this investigation, you will use gel electrophoresis to make a DNA fingerprint of each suspect, the owner of the bicycle, and the DNA from cells on the hair. You will then compare the DNA fingerprints to determine to whom the hair belongs.
Okay, so we set up the problem. Guys, mountain bike got stolen. There's some evidence DNA, a hair that was left behind. That hair has cells, it has DNA. We're going to extract the DNA. We're going to run it through a gel to kind of get a fingerprint of that hair. Whose hair is it? We have two suspects, so we're going to get a court order, go get their blood, get some of their cells, get some of their DNA, and run it as well to see who's going to match that evidence DNA. And then we're also going to run the victim's DNA to make sure it wasn't his hair, and also a DNA ladder, a DNA standard to kind of give us an idea of how big these pieces of DNA are going to be. We're going to cut the DNA with restriction enzymes. Guys, that's something you should have read about in your book, enzymes that cut DNA at very specific sites. So guys, let's go ahead and go to our next step.
Explore the lab to learn what is available for your investigation. You must click on each item in the checklist. Once you are ready to move on, click Procedure.
Okay, so we're going to click on each one of these items. First, Aragos gel, this right here. Let's go ahead and click on that.
Agarose gel. Use the wells in the agarose gel to load DNA samples. Smaller fragments of DNA travel through the gel more easily than larger ones, and, over a certain length of time, travel farther.
Okay, next we're going to do DNA marker, sometimes called a DNA ladder.
DNA markers. DNA markers are DNA fragments with known lengths.
Now we're going to click on our digested DNA sample. So these are DNA samples right over here. Of our suspects 1 and 2, the victim, and the evidence DNA, the hair we found on the crime scene. We've cut the DNA with enzymes as well.
Digested DNA samples. 4 DNA samples have already been cut into fragments by restrictive enzymes.
Now, that's kind of key. Remember, everybody on this planet pretty much has different DNA. No one's DNA is exactly the same unless you have an identical twin, which means if you cut DNA with restriction enzymes, no one's DNA should really be cut exactly the same. So that's kind of the key thing here. Electrophoresis chamber, that's our next one.
Electrophoresis chamber. Electrophoresis takes place in the electrophoresis chamber.
Okay, so that's going to be what's going to run some of the power through our gel along with the power supply. So let's go ahead and click.
Power supply. Use the power supply to produce an electric current in the electrophoresis chamber.
Next, bromophenol blue. That's kind of our stain. That's going to stain our DNA and kind of help it pop from the gel so we could actually see it.
Bromophenol blue. Use the bromophenol blue to see how far the DNA samples have moved. Bromophenol blue is a dye that travels with the DNA samples in the gel.
TBE buffer is next. This is a buffer that helps the electricity current run through the gel.
TBE buffer. Use the TBE buffer solution to help carry an electric current. TBE buffer is added to the gel in the gel box.
Then we have our gel staining tray where we're going to put our gel, kind of stain it with a stain to kind of help that DNA pop.
Gel staining tray. Use the gel staining tray to hold the agarose gel for staining after the DNA fragments have been separated by electrophoresis.
Next, we have ethidium bromide, a really nasty substance, a carcinogen, but it helps actually stain our DNA. It intercalates the DNA, gets in there, and it's going to help it fluoresce once we run it in a UV box, once we shine some UV light on it.
Ethidium bromide. Ethidium bromide is a fluorescent dye used for staining DNA.
Tool you're familiar with, a micropipette, which is going to help us load our samples into our gel over here.
Micropipette. Use the micropipette to transfer dye and DNA samples.
Then we have our micropipette tips, which, guys, you know better, they should always be inside a box and closed, but this is a virtual lab, so they have them out for you.
Pipette tips. Use a clean pipette tip before transferring dye or DNA samples to avoid contaminating the samples.
Then we have some deionized water. We always use everything when we do a lab like this. We're using deionized water to clean everything, to help us stain things and wash things away.
Deionized water. Use the deionized water to rinse the gel after it is stained with ethidium bromide.
And then last one, our ultraviolet box. That's going to help us kind of see our DNA fragments, see our DNA gel.
Ultraviolet light box. Use the ultraviolet light box to help read the gel after it is stained with ethidium bromide.
Okay, so we've hit every single one of these. We are ready for our procedure.
In this lab, you will produce a DNA fingerprint from cells on a strand of hair found at a crime scene. You will compare the DNA fingerprint of the evidence to the DNA fingerprints of the victim, suspect 1 and suspect 2. Open the lab notebook and explain how comparing DNA fingerprints can help identify a person who has committed a crime. Once you have written your explanation, click the arrow to advance to step 2.
Okay, so this is where on your own pieces of paper, you want to kind of write your answer to this question. So I'm going to open up the lab notebook, and it says, explain how comparing DNA fingerprints can help identify a person who has committed a crime. So on your paper, you could write your answer there. I don't want to give this away, so I'll just type something in there. So go ahead, you could pause the video here so you could record your answer. If you're having trouble, I'll give you a little bit of help. Guys, remember DNA fingerprints, they're gonna be different for everybody. So whoever's DNA fingerprint matches the evidence DNA fingerprint, you're gonna know that that person, possibly that's the hair that it belonged to. So possibly they're the one who maybe committed the crime or has to at least explain why their hair was found at the crime scene. So I'm going to go ahead and go to our next step.
The agarose gel is in the electrophoresis chamber. Click the TBE buffer to cover the surface of the gel with buffer. Once you have covered the gel with buffer, click the arrow to advance to step 3.
Okay, so notice our gels here. We have the wells right here. That's where we're going to put our DNA. But first, let's put some buffer over it. And once again, that buffer is going to help the electricity current run it through this gel. Notice we have a negative end here and a positive end, but let's go to our next step.
Click the micropipette to load the bromophenol blue dye and mix it with the marker DNA. The mixture is then ejected into the marker DNA well. Once you have filled the marker DNA well, click the arrow to advance to step 4.
Okay, so what we're going to do is we're going to load the DNA marker first. We're going to put some stain in it first to kind of once again help the DNA so we can see it go through the gel. Usually the marker already has it, but in this case, we're going to put it in there. Give our micropipette tip, put the dye, and then we're going to load it on this first well right here. Be careful, don't poke the gel. We've loaded our sample. And now we're ready to load the other samples.
Click the micropipette to load bromophenol blue dye and mix it with the victim's DNA. The mixture is then ejected into the victim well. Once you have filled the victim well, click the arrow to advance to step 5.
So there, now we just have the victim's DNA. So once again, we have the victim's DNA. We've cut it with a restriction enzyme. We're using the same restriction enzyme to cut all of these. And once again, they cut at very specific spots. So we've done victim 1. Now we're going to do suspect 1, suspect 2, and also our evidence DNA.
Click the micropipette to load bromophenol blue dye and mix it with suspect 1's DNA. The mixture is then ejected into the suspect 1 well. Once you have filled the suspect 1 well, click the arrow to advance to step 6.
So we've loaded suspect 1. So far we have our DNA ladder, our marker. We have our victim. We have suspect 1. Now we're going to do suspect 2.
Click the micropipette to load bromophenol blue dye and mix it with suspect 2's DNA. The mixture is then ejected into the suspect 2 well. Once you have filled the suspect 2 well, click the arrow to advance to step 7.
So now we have suspect 2 on this well right here, and now we're going to do our last one.
Click the micropipette to load bromophenol blue dye and mix it with the evidence DNA. The mixture is then ejected into the evidence well. Once you have filled the evidence well, click the arrow to advance to step 8.
Okay, so notice we have our gel complete. Once again, we have our DNA ladder, our marker. We have our victim right here, suspect 1, suspect 2, and this is the evidence DNA, that hair that we found at the crime scene. We got some cells, some DNA out of there, cut it with some restriction enzymes.
Click the power supply to connect the electrodes and produce electric current for 1 hour. Notice that the negative electrode is closest to the wells, and the positive electrode is farthest from the wells. Once an hour has passed, click the arrow to advance to step 9.
So notice we've connected our current, once again, negative here, positive here. Remember, DNA is negative, so it gets repelled here. It's attracted to the positive, so it forces it to go through this gel. And remember, that gel is going to separate these fragments of DNA by their size. Larger pieces have a harder time going through the gel, so they don't go very far. Smaller pieces are going to be able to go through a lot, are going to be able to go through it a lot faster, a lot further down the gel.
Drag the gel from the electrophoresis chamber to the staining tray. Click the flask of ethidium bromide stain to flood the gel and allow it to stand for 30 minutes. Click the staining tray to discard the stain. Once you have stained the gel, click the arrow to advance to step 10.
Okay, so let's grab our gel, and does it go in the sink? No, it doesn't go. The ultraviolet box? No, that's silly. It goes in the staining tray right here. Next, we're going to get the ethidium bromide. That's going to stain the gel. That's going to make the DNA pop visible. We let it sit there for about an hour. Then we are going to discard the ethidium bromide. Now, this is a big no-no. You never pour ethidium bromide down the drain. You actually hold it in a special container, and then there's a company that comes and picks up that ethidium bromide because, once again, it's a cancer-causing agent. So we've stained it. Now let's go ahead and do our next step.
Click the deionized water to rinse the gel and allow it to stand for 30 minutes. Click the staining tray to discard the deionized water. Once you have rinsed the gel, click the arrow to advance to step 11.
So now we're going to discard this. Once again, we wouldn't do it down the drain. We would hold it in a special container.
Drag the gel to the light box and click On in order to read the gel. Open the lab notebook and record the estimated size of each band of DNA. To determine the length of the evidence DNA, the victim's DNA, and the DNA from both suspects, compare it to the marker DNA. Once you have recorded your observations, click the arrow to advance to step 12.
Okay, and we have our results. So notice we have, once again, these are DNA bands. These are fragments of DNA. Once again, we cut our DNA with a restriction enzyme. And guys, notice pieces up here, they're huge. They're big. They don't go very far through the gel. Pieces down here are smaller. They're able to navigate through that gel a lot faster and go a lot further down. So notice, just looking right away, hopefully you're like, oh, I think I know who committed this crime, who possibly sold this bike, or at least has to explain why their hair was found on the crime scene. And hopefully you see that it's suspect 2. Notice how suspect's 2 DNA matches perfectly with the evidence DNA. So it looks like that right there is our we pretty much solved this possible crime right here. Once again, suspect 2 matches perfectly. Notice here's our marker. Victim really doesn't match. Suspect 1 really doesn't match as well. Now we're going to open our notebook, because what we have to do is we have to estimate how big these bands are. So the marker one, pretty easy. That they already tell us what those numbers are. That first band is 23130, 21,130 bases. That's how big that fragment is. Then the next one right here is 9,416. Then we have our next one, which is 6,557. Then we have our fourth one, 4,361. And you guys could record these on your own paper. Then we have we have 2,322. And then our last one, 2027. So we have 6 bands. Let's just make sure 1, 2, 3, 4, 5, 6. We recorded these, and now we could estimate how big the victim's DNA bands are by comparing it to the marker. For example, this first one right here, it looks like it's a little bit more than this one. So I'm going to say it's about, 2,400. That's my best estimate. Then this band right here is right below this one. So I'm going to say it's about, I don't know, maybe 1,900. And then this last one's pretty much down here. I'm going to say that's 1,000. Okay, now let's look at suspect 1. Notice its DNA, it has one here that's almost like a perfect match to the marker. So I'm going to say that's 9,416. Then this one, it's below it. So I'm going to say it's about maybe, I don't know, maybe 8,700. Now your numbers can differ. Maybe you're seeing it slightly different. It's fine. Just record your numbers. And then we have this one that's a little bit below this one. So I'm going to say it's about maybe 4,000. Now we do suspect 2. Notice suspect 2, this one's slightly below this one. So I'm going to say it's 2,200 or 22,000. Then we have this one right here that's pretty much spot on to this marker here. So I'm going to say it's 6,557. Then this one's kind of below it. So I'm going to say, that's tough. I'm going to say maybe 5,200. And then the last one is down here. It looks like it's slightly below. I'm going to say 2,000. And notice the evidence DNA is a perfect match, so I'm just going to record the same numbers. Okay, so we have our estimates. Once again, you guys could do this on your paper. Guys, you could pause it right here if you need to catch up, but I'm going to go ahead and kind of click our next step. So let's go ahead and see if we could do our last step.
You are now ready to analyze your data and draw your conclusions. Open the lab notebook and answer the questions.
Okay, so now this is where you could answer these questions on your own paper. So question number 1 says, evaluate. Does the DNA found on the hair match suspect 1, suspect 2? Hopefully you know, oh, that's definitely suspect 2. Question number 2 says, why do a series of bands appear on the gel? Well, guys, that's something we did in the lab. Hopefully you're able to answer that. Hopefully you know that, okay, well, first, We've cut the DNA into fragments, and then we were able to stain the DNA that kind of makes it pop, that makes it visible. That's why we see these bands on this gel. So I'll just put the cut with restriction, enzymes, and stain DNA. It says, why is the largest DNA fragment band found closest to the well in which it was placed? Well, guys, remember, large pieces don't travel very far, so you could just put, large pieces or fragments don't travel far. It says, what is true of DNA fragment band closest to the positive end? Well, guys, the positive end is over here. The negative end is over here. So if it's positive, if it's really close here, what does that tell you about the size? That they're small fragments. And then we have predict what would happen if the electrodes were plugged in the wrong outlets. Let's say we actually had the positive one up here and the negative one here. Guys, what do you think is going to go wrong? You know what? I'll let you kind of answer that one. I don't want to give you every single answer, so I'm just going to put try your best. Just think about that. Okay, once again, DNA has a negative charge. So if you have a positive end on this side, what's going to happen? And then last one, why is DNA fingerprinting more conclusive when proving a person's innocence rather than their guilt? Guys, this one's a thinker. You know what? I'm going to let you think. Think away. Guys, don't freak out. I know some of you are like, oh, Miss Rhea, I'm getting a headache. That's not a headache. That's you thinking for the first time during distance learning. Guys, see if you could figure out why would it be more conclusive to prove a person's innocence rather than guilt. I'm just going to try your best. So, guys, hopefully you filled out all these questions on your own handout. For those of you who actually do have access to this, to print this, actually not print it, you want to do a PDF, save it as a PDF, just go to print. And you're going to see that all your answers are on here, and you want to save as PDF. Now, some of you might have a printer on there, but just click on there, put save as PDF, and save it to your desktop, save it to a flash drive, save it somewhere, call it something like your name, and then Joe Electrophoresis or DNA Fingerprinting Lab, and then you could save it on there. So, guys, that pretty much finishes our actual lab. So let's see if there's anything else. Let's say we are done. And yes, congratulations, we completed the investigation. Guys, hopefully you found this useful for those of you without Flash. Guys, if you have questions, if there's something you didn't understand, guys, send me a message on Edmodo, send me a message on Instagram, send me an email, request a Zoom meeting with me, or ask it during one of the Zoom meetings, and I'll make sure to try to answer those questions and get you guys comfortable with Joe Electrophoresis. Guys, today is Wednesday, the 15th. Don't forget, tomorrow on the 16th, we have our Quizzes mid-week knowledge check. So don't forget about that. Well, guys, it's awesome. Hopefully you found this investigation pretty awesome and very educational, and that you actually now know a little bit more about electrophoresis and DNA fingerprinting. Well, guys, have an awesome day today.